Good day, this is the Professor, CBMG PhD, and today's Sunday, August 6, 2023. It is 4:07 and 4 seconds, AM Eastern Standard Time, and today's research, Bioinks. So let's jump to it here. Enhanced tumor modeling using lapinite, Bioinks, for 3D bioprinting. Date August 3rd, 2023. Source: Terasaki Institute for Biomedical Innovation. As scientists have developed a nano-engineered bioink with improved bonding and cross-linking capabilities for 3D bioprinted tumor models. These capabilities are enabled by the addition of lapinite, highly charged, disc-shaped nanoparticles that enhance and retain the biological signaling that occurs in the tumor microenvironment so that more accurate tumor models can be created for study in anti-tumor drug development. As tumor microenvironments are complex with the support of connective tissue matrix containing multiple cellular components such as tumor cells, immune cells, organ-specific cells, and collagen-producing, cancer-associated fibroblasts. In addition, there is an extensive cell-to-cell physical interaction and interactive signaling via a variety of biological factors that are difficult to model in an accurate and representative manner. The use of 3D bioprinting offers a versatile approach for creating in vitro tumor models where precise 3D tissue structures can be created. To create an in vitro model, the printers use a cell-laden biomaterial solution termed Bioink, custom-made for the intended tissues. The bioinks in this study were comprised of cancer and normal cells from the tumor microenvironment, which were embedded in a biocompatible gel, typically a type of polymer. This cellular gel mixture must be optimally formulated to exhibit mechanical and biofunctional properties that accurately recapitulate the tumor microenvironment. In order to achieve these properties, the research team chose gelatin methyl oil as a polymer base for their bioink, a biocompatible material with tunable properties for structural stability and porosity with binding sites conducive to cellular adhesion and survival. Adding lapinite to the methyl oil not only created a reinforced network within the bioink, but also improved printability by conferring shear, thinning properties, the ability to deform under stress, and then quickly self-recover. In additional tests of their new composite as a biomaterial for the fabrication of a tumor model, the scientists made different methyl oil lapinite formulations and used them to encapsulate human pancreatic cancer cells. The formulations, which gave optimum cell viability, were identified and used in creating 3D bioprinted multicellular tumor models. These multicellular models were made by adding fibroblasts, cells which are pivotal to pancreatic cancer tumor progression, to the methyl oil lapinite pancreatic cell bioink. Tests of these models showed that the cells had good viability, particularly with higher lapinite concentrations. As furthermore, higher concentrations of lapinite were also observed to increase the size and distribution of co-aggregations of the two cell types. This more fully depicts the native pancreatic tumor structure and its promotion of cell-to-cell interactions. The team went on to study the effects of lapinite on the gene expression of various biomarkers which are promoters or indicators of tumor progression in pancreatic cancer. They found that increased lapinite concentrations unregulated production of tumor cell growth factors, as well as remodeling and cell differentiation genes by 10 to 20-fold. The effect of lapinite on gene expression was even more pronounced on the fibroblasts, with overall unregulation of the gene pools, especially for genes related for growth factors, which were increased up to 60-fold. As fibroblasts play an important role in pancreatic tumor progression via signaling by these growth factors, this is a significant demonstration of lapinite's influence. The improved signaling between cancer cells and fibroblasts by released biological factors also increase the expression of genes that are related to the cell cycle, specifically genes that indicate a decrease in the proliferation of cancer cells. The decrease in the proliferation of cancer cells is known as cancer dormancy, which is one of the factors affecting resistance to chemotherapy and cancer recurrence after treatment. The scientific investigators and microbiologists say the study of the effects of laponite on the 3D printed tumor model has shown that not only does laponite improve mechanical characteristics of the model, but it can also selectively influence the biological signaling that are a part of tumor progression. And this gives us the versatility to recreate more accurate tumor models of different types so that targets for effective therapeutic drugs can be identified. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.